Hello dear students. In last class we studied about the process of cell cycle. How a cell undergoes different phases before dividing. Today we will study about the first type of cell division, which is called as mitosis. Before going into the details of the process of mitosis, let us first see what are the general characteristics shown by the process of mitosis. So, this process of mitosis is called as an equational division. Very important. Many a times a question is asked, why mitosis is known as an equational division? The reason is that the number of chromosomes present in the daughter cells after mitosis remains the same as present in the parent cell. If the parent cell is diploid, that is 2n, the number of chromosomes after the division in the daughter cells will also remain 2n, that is diploid. But if the parent cell is haploid, that is n, after division, the number of chromosomes in the daughter cell will also remain n. So we can say that this mitosis can take place in both haploid as well as diploid cells. So it is called as equational division. Now next characteristics is, after every mitosis, two daughter cells are produced. One mitosis will give rise to two daughter cells. That is called doubling of the cell. So in bacteria, it is generally mitosis that helps in replication. So that's why we say doubling. So two daughter cells are produced after each mitosis. Now let us now go into the significance of mitosis. It is so important. Let's see. Number one, it is responsible for our growth. After the birth, the organism grows only by mitosis. Whatever growth takes place in any organism, it is because of mitosis. The number of cells keep on increasing. As a result, the wholesome growth takes place. Second, repair or healing. Every day, there are so many wear and tear shown by the different cells in the body of any organism. So that repairing, that healing is also done by mitosis. And third is asexual reproduction. All asexual reproduction, be it unicellular organism or a multicellular organism, it is because of mitosis. So these were the characteristics of the process of mitosis. Now let us go into the details of the process. How a cell undergoes the process of mitosis and produces two daughter cells. Let's study it. Now let's study the process of mitosis. You can see a flow chart. Here, this is the interphase. Interphase, as I already told you during the cell cycle study, it is the resting phase from the point of view of cell division. The cell is preparing itself for the next division. After the interphase is complete, then this cell is ready to undergo mitosis. So this is a cell after interphase is completed. Here I have written 2n is equal to 2, which means it's a diploid cell having two chromosomes. Now, during an interphase, what is the property of a cell? A cell has got a nuclear membrane. You can see this is a nuclear membrane. There is a nucleolus. This structure is a nucleolus. And the genetic material is in the form of chromatin, or you can say it is a thread-like, interwoven thread-like structure here. So in this way, the cell cannot undergo cell division. It has to show many changes, and that changes is done in two stages. One is in karyokinesis, where the nucleus is undergoing division, which includes prophase, metaphase, anaphase and telophase. After telophase is completed, then there is cytokinesis, division of the cytoplasm, and finally this cell develops into two daughter cells. One cell divides into two daughter cells which are exactly like the parent cell. Okay, so you can see that in the interphase there is a nuclear membrane, there is nucleolus, the genetic material is in the form of chromatin threads, and these are the pair of centriole which will help in the formation of esterase and spindle fibers. Okay, and now we will study what all changes this cell undergoes to produce two daughter cells. So let's study this karyokinesis and cytokinesis in more detail. Let us now study the process of mitosis in detail. Here, this is the cell in the interphase. In the interphase, when the cell is ready to undergo karyokinesis, it has got a well-developed nuclear membrane and nucleolus. And the genetic material is in the form of long, very thin fibers called as chromatin. When it enters the first phase, that is prophase, it shows certain changes. What are the changes? First, the nuclear membrane starts disintegrating. Nucleolus also starts disappearing. And this chromatin shows coiling and condensation and gets converted to chromosomes, which are more thicker. Okay, and you can see these are a pair of centrioles. They start moving to the opposite poles. These centrioles will be responsible for forming the spindle fibers. Here you can see. So after prophase, prophase is the longest phase of all these four phases. Okay. After prophase, next stage is metaphase. It's not a jump from prophase to metaphase. In fact, there are sequential changes. Okay. So in the metaphase, you can see the spindle fibers are completely formed, and the chromosomes, they get attached to these spindle fibers with the help of kinetochores. Also, these chromosomes, they split lengthwise and remain attached at the centromere, and these arms are called as chromatids. Metaphase is the shortest of all these four phases, but it is very important from the chromosomal studies point of view, because all chromosomal studies under the microscope are done in metaphase. Why? It is because all the chromosomes in the metaphase are present at the center of the cell, which is called as equatorial plate or metaphasic plate. So you don't have to search for the chromosomes here and there. They are all lined at the equatorial plate. Also, these chromosomes are very thick, quite visible. That's why the photograph is clicked and then they are arranged properly. This process is called as karyotyping. Karyotyping is the process of study of chromosomes during metaphase stage of the cell division. And the pictures are prepared. Those pictures are called as ideograms. Okay. So, in the metaphase, the chromosomes are at the equatorial plate. Now, the third is anaphase. In the anaphase, you can see the cell has stretched itself, and these chromatids, they have separated from each other and they start moving to the opposite poles. These centromeres are moving very fast and the arms are following them. So they give the appearance. In previous chapter I told you different types of chromosomes during the anaphase. You must be remembering metaphasic, submetacentric, submetacentric, acrocentric and telocentric. Those chromosomes are studied here during the anaphase. So you can see during anaphase the cell stretches itself. Now these chromosomes, chromatids, they have separated in the chromosomes and they start moving to the opposite poles. This is also a very important stage, because if this separation is not taking place, then there will be abnormalities in the cell. Okay. Now next, last part of these 4 stages is the telophase. Telophase is the reverse of prophase in a way. You can see what's happening. The nuclear membrane starts reappearing. Nucleolus also starts reappearing. And there is uncoiling of the chromosomes. And the spindle fibers, they start disintegrating. And as a result, you can see the formation of 2 cells taking place. Along with the telophase, cytokinesis also begins. And as a result, there is a separation of the cytoplasm. There is the furrow formation towards the center of the cell takes place. As a result, the 2 cells in animal cells, they pinch off and they develop into 2 daughter cells. Whereas in case of a plant cell, this pinching is not possible because there is cell wall. Therefore, in them, there is a cell plate formed from the endoplasmic reticulum. And then that cell plate is laid on both the sides by the middle lamella, and ultimately 2 cells are produced. Okay, so after telophase, cytokinesis will take place, the cells will separate and 2 daughter cells are produced. And you can see these daughter cells which are produced, they are exactly like their parent cell. That's why it is called as equational division. And you can see after this division, every division results into 2 daughter cells. So this was the process of mitosis. It is very important. Please go through the different stages. You must know what all processes happen at different stages. And please practice the diagrams also. Significance I have already told you. Mitosis is very important for the growth of any organism. It is important for healing and repairing. Also in many organisms, the asexual reproduction takes place through mitosis. There is also one very harmful side of mitosis also. You must have heard about cancer. What is cancer? Cancer is not a disease. It's a disorder. It's a disorder of mitosis. Because mitosis has to be controlled. Just imagine if there is no control in the mitosis, the cells will keep on producing more and more number of cells. And those number of cells, they will aggregate to form the tumor. So uncontrolled mitosis is called as cancer. That is tumor formation. But in our body, there is a mechanism to restrict the number of mitosis after a particular stage. And as a result, mitosis is under the control. It is under the genetic control. And our body produces only those number of cells which are required. So I hope you have understood this process of mitosis well. Thank you.